Welcome back to Bio 255 Microbiology Lab. This is session 3, so please have your lab manual open to session 3, page 1. Today's lab is entitled Aseptic Technique. We are going to learn how to inoculate and subculture bacteria. That means we are going to transfer bacteria from a pure culture, known as a stock culture, into a new sterile medium, and we're going to maintain the purity of that culture. A pure culture means only a single species of bacteria is actually present. So we need to use aseptic technique to prevent contamination of our newly inoculated culture. Before we begin, let's use a couple of aseptic techniques we learned in previous sessions. Number 1, wash our hands. Follow me to the sink, and let's recall that in this class we use, unless we have a spill, we use mild hand soap as a simple surfactant to physically remove surface contaminants. We know there's a lot of surface contaminants. We saw that in the last session with our fingertips before washing plate. So I'm going to wash my hands, and after I do so, I want to talk about why I'm not going to wear gloves today. We do have boxes of gloves in our micro lab, and sometimes on a few occasions in this course I will wear gloves. When I do so, it's to prevent exposure to certain chemicals or when I'm working with a fairly serious pathogen. But for the most part, we don't want to wear gloves because without gloves, we can always feel when we have spilled bacterial cultures on our skin, and that sensation of contact is what causes us to immediately disinfect ourselves. If we spill and we have gloves on, we may pay no attention to that spill, and then we proceed to move that bacterium to many different tools, equipment, surfaces, and potentially to other parts of our body. So we are not going to wear gloves for the most part in this course. That being said, our mantra will always be, do not touch men. Men, M period, E period, N period. That means under no circumstance in this course do you ever bring your fingers up to your mouth, your eyes, or your nose. People do this an awful lot, don't they? They bite their fingernails. Unfortunately, they pick their nose. They're constantly wiping their eyes. They're going like this, picking their teeth. Those are the opposite of aseptic technique. And in this world of pandemic, you never want to bring your hands near your face. So let's go back to the bench, and let's go ahead and let's disinfect our bench. If you recall, we use 10% bleach, and we squirt it low over the surface of the lab bench, and then what do? We take a couple of dry paper towels, and we spread that bleach across the surface to cover everything, and we simply let it air dry. Okay. So now we're ready to take a look at our learning objectives for the day. As far as what my expectations are for you in lab session 3. First of all, I want you to be able to use aseptic technique to inoculate and subculture bacteria, as we just stated. I also want you to use and get familiar with both broth culture and agar media. So broth and agar media, and being able to move easily between one and the other. And we are going to evaluate the effect of temperature on the growth and culturing of a given bacterium today. And we're going to take a look and see if it might sometimes affect colony morphology. In this course, I expect you to be able to speak the language of microbiology and write the language of microbiology. Only by doing both do you become completely fluent in this discipline. So every time that I teach you a new bacterial genus and species nomenclature, I'm going to expect you to be able to spell it and to pronounce it. So today we are going to work with a bacterium known as Serratia marcescens. That's Serratia marcescens, and we'll talk more about it in a few minutes. But that's something that you should be able to pronounce. and spell. And I realize it's Latin because in microbiology we do not have common names for bacteria. So you look at something like Rana pipiens, the green frog. We can call it a green frog. We can't call a bacterium a green bacterium. We always use the binomial, the Linnaean binomial, which is the genus capitalized and the species lowercase, all lowercase. And they are either italicized if they are written or they are underlined if they are printed. So we'll look at that. And I want you to be able to use binomial nomenclature properly and write it out in proper scientific format before the end of this session. And finally, bear in mind that in this course, always, lab safety comes first in everything. So today we are going to identify some safe laboratory behaviors. And I want you to know those and own them. We are going to start with the fact that I'm sitting down. Whenever I do culture transfers, I am always sitting, and you must always be sitting. When you do culture transfers, you have to work with a flame. So the first thing you do is you pull your hair back in a scrunchie if you have long hair. If it's short, you're fine. If you are working with that open flame and you have glasses, you are not exempt from wearing goggles. Glasses do not have the side shields or the bottom shields or top shields of goggles. We have clean goggles. They are disinfected goggles. And I'm going to show you that goggle cabinet when we clean up today and we put them away. For now, I'm going to show you that they fit on top of my glasses so I can wear both comfortably while I work in the lab. What other safety procedures do we need to bear in mind? We saw in session 1 that the inoculating loop is a major tool, and we are going to use that loop today. And we also said in session 1 that we had to flame it to sterilize it. When you flame a loop, it gets very, very hot. So hot that it will burn right through the skin if you touch it. So when you work with your inoculating loop or needle in this course, when you are done after you have sterilized it, you place it on the lab bench. You never hand it to another person. If you are transferring it to your lab partner, it goes down on the bench and she or he picks it up from the handle. Okay, let's see. Also, two other safety things. Recall that we have test tube racks and we have tubs for our glass test tubes. And glass test tubes sit very safely in a rack or in a tub. But recall that I said you never, ever, ever lay a tube down on the bench like this, okay? So we have to keep that in mind. And that begs the question of, what if you do actually spill? What if you tilt a broth tube like this too much and it falls out onto the lab bench? What do you do? Or onto your hand? The first thing you do whenever you suspect that you have spilled is you scream, "Stop! I spilled." It loud enough so that everybody in the lab can hear you. And what does everybody else do? They stop. They stop what they're doing. If they're walking across the floor, they stop in mid-motion. And the reason for this is we need to assess, more importantly, the lab supervisor, in that case me, needs to assess what you spilled and where it went. The next thing to know about a spill is that if a spill occurs and I hear a student say, "Stop, I spilled," everybody stops, and I need to be able to speak with that student without any side conversations going on. So you not only stop physically, you stop speaking. And then I will say, "What did you spill?" And they might say, "I spilled Serratia marcescens." And then I know what I'm cleaning up. Where did you spill it? On the lab bench. Is it on your hands? No. Did it splatter? Yes. How far did you see it splatter? It's on my pants. So I need to have that conversation with any student that spills. And then the next thing you need to know is that I am the only person in this lab that ever cleans up a spill. You never clean up your own spill. So you say, stop, I spilled. Marla, come and help me. I will be there in an instant and I will clean up that spill. I know how to clean up that spill and clean up that spill safely, but I also know how to clean up you. So we will, depending on what you spill and how widely it has splattered, I will take different precautions. Okay, nobody in the room will move again or speak again until I give the okay. So that's our spill safety instructions and protocol. I never get upset with a student who spills. Everybody in this classroom is going to spill at some point. The only time and the only reason for getting upset with anybody who spills is if they do not report it. Because if you don't report it, you're endangering yourself and others. You are contaminating the room, and we need to keep it as decontaminated and as aseptic as possible. So that's the deal. Plus, we always learn by everybody's mistakes in this course. You make a mistake, hey, we hail it the first time at least, because then we use it as an example and show other students what not to do so that we all don't follow in your shoes and make that same mistake. Okay, so let's see. That is what we do in case of spills. I think we are ready to begin. First thing that I want to do is I want to show you how to light our flame. So if we can zoom in here, I am going to draw your attention to the gas jet over here. Now, the gas jet is a blue nozzle that says gas on it. When it is in this horizontal position, it is off. I have to actually physically lift the cap and rotate it so that it is perpendicular with the base in order to get gas to come through the microburner. And you can probably, if we're really quiet, you can hear that gas come through. That's not something we want to let happen for very long. So within a second or two of turning the gas on, you want to use your striker. And remember that the striker has a flint on it that moves across the rasp and ignites the gas. Now, I'm going to go ahead and I'm going to turn this off again. I'm going to turn it back on. We're going to do this again. I want you to note what I did. I was in my chair, where I belong if I'm going to ignite gas. My head is way back away from that very large flame. My hands are out, almost at an arm's length. In fact, an arm's length is a good length at which to ignite a microburner. I'm holding the cap to just to the edge, down over the gas, and I'm making sure that I can light that gas in one strike. So let me try this again. I'm going to turn on the gas. I'm going to put arm's length, one strike, one spark ignites the microburner. The microburner has a couple of regulatory dials here. Let's take a look. Underneath, I can change the amount of gas flow through the microburner by rotating this knob. And so I can increase the gas coming through, or I can decrease the gas coming through. I'm going to set it down a little bit lower, just about like that. Hopefully you can see on the video that there is a light blue series of cones over the mouth of that burner. And that is actually in the light blue flame, the hottest part of the flame. When you see, I'm going to open this up a bit, when you see orange flame like that, that's not the complete combustion we want. And so we don't need to have an orange flame on our burner. The problem with not having an orange flame on the burner, safety-wise, is that this light blue flame you don't always see. So in this lab, we have to assume every burner, if it is out on a table, is actually on. And be respectful of it. The burners should not be at the edge of the bench. They shouldn't be towards you, right up beside you. They should be roughly in the middle of the bench, an arm's length from everybody who is sitting down. If you have it at the edge of the bench and people get up to walk, they might accidentally move their hand or flick their ponytail through that. So we don't want to do that. Always have it in the middle of the bench. Remember that I mentioned this bottle, this ethanol bottle, should be nowhere near the flame. And the other thing I wanted to show you is that we can adjust the combustion by allowing less or more oxygen in. And that's what this gappy dial is for. It allows less oxygen in, or you can open up these windows to allow more oxygen in. So I'm just going to leave it open because right now it's in pretty good shape, the way I want it. Let's take a look at our loop. When we flame a loop in this course, the first time you flame it, I generally just very, very briefly run the handle through. But I don't want to run the handle through where I'm touching it because that's going to burn. And if you keep the handle in the flame, it's going to run right up this metal and it's going to burn your hands. So just a very quick decontaminating run through for the handle. But when we look at the nichrome wire, what we do want to do is we want to start up where it connects to the handle, and we want to completely sterilize that wire. And it turns bright orange. And I hope you can see this with the video, but you can see that bright orange. When I put this loop right over the blue, it immediately turns orange. That means it's instantly sterilized, okay? So we don't have to hold this in the flame forever. In fact, if you do, you weaken the wire. So let's just decontaminate it, or excuse me, sterilize it like that. Now remember, that is red hot, and it will melt plastic. If I touch this plastic, it's going to melt it. If you need to, for whatever reason, immediately cool off your loop, say you just finished your final transfer and you're going to put the loop back in your drawer. You don't put the loop back in the drawer red hot. So if it just came out of the flame and you want to cool it immediately, you just simply run it under water for a split second. There it is. Now look, I can actually touch it and it's not hot at all. So because of the heat capacity of metal, it heats up fast, but it also loses its heat fast. We can simply run it underwater if we want to put it away. So remember, when you are done, after you've done the transfer and you heat it and it's red hot, what do? You put it on the lab bench. You do not ever hand a loop in person to anybody else. It goes on the lab bench for them to pick up from the handle if they need it. All right, so let's take a look at today's lab session. We are ready to do our culture transfers. Every time you work with live bacteria, you will have what we call your stock cultures. Your stock cultures are pure cultures that I have prepared ahead of time of the bacterial species you will use for the day. Today, our bacterial species is called Serratia marcescens, and each of the 2 stock cultures I have here are labeled as such. Serratia marcescens is a ubiquitous bacterium. That means it's distributed just about everywhere in the environment. It's even in animals and plants. It is found in water, in soil. It causes infection in humans once in a while, usually in immunocompromised individuals. We have to be careful. We have to use aseptic technique to work with this. I want to point out this stock culture. This is nutrient broth. In nutrient broth, or any broth medium, if there are bacteria growing in that broth, it becomes turbid. Turbidity is a term we use to mean cloudy. And you can see, if I mix this up, just how cloudy it is, especially when compared to a sterile culture tube of nutrient broth. And you can see the tremendous difference. Now, just because this broth tube is clear doesn't mean that it is necessarily sterile. There may be several bacterial cells in there that we just can't see. However, I know it's sterile because I autoclaved these before class. So this is sterile media. This obviously has bacteria growing in it. If you ever pick up a cloudy or turbid broth tube, be aware that a single drop of that culture contains trillions of bacterial cells. The other stock tube that we will work with of Serratia is a nutrient auger slant. The bacteria are growing on that slant, and we will use that as well. Okay, for today's lab, your lab manual asks you to obtain 4 sterile nutrient auger tubes and 4 sterile nutrient broth tubes, which I have in this rack here. I want you to note that there are caps on these tubes, but when I picked the tubes up, I did not actually pick them up by the cap, did I? When you work with glass tubes in this class, you never ever pick them up by the cap. Caps are loose, and they are not meant to be airtight. They do not form a seal. They actually should be fairly loose around the top rim of the tube. You can push them down and make them a little tighter, but that's not the way we want a cap. We want it sitting loose. It allows oxygen into the culture if oxygen is needed, but it is never meant to be a seal. So please remember, whenever you pick up test tubes, you grab them by the glass and thus securely. Okay, also when you've got tubes in a test tube rack, look at how wabi-sabi loosey-goosey they are. They are constantly tilting. So when we do transfers between glass tubes, we do not leave the tubes in the rack to do the transfer. We always hold the tubes up in our hands and we transfer from a stock to a new sterile medium. We do this for several reasons. We don't want to move bacteria across a lab bench. We want to move bacteria from the stock tube to the sterile tube in as short a distance as possible. And so if you have both tubes in one hand, grasping tight, you can do this very efficiently and without the risk of contaminating other areas of your workspace. Also, be aware, please, that when you do culture transfers, you are the only person involved. You do not ever allow your lab partner to handle a tube while you transfer live bacteria into that tube. Let's have a look what can happen if you are not in control of the tubes. If I put a stock tube of Serratia marcescens here, and I wish to transfer it into this tube, and I flame my loop to sterilize it first, and then I try and get into the tube, I actually can have trouble and struggle with that, contaminating my loop. Or once in, when I grab the bacteria off the tube and I move over to the new medium, if I go in and I miss, what have I just done? I've just contaminated the outer rim, or potentially missed altogether and contaminated the test tube rack. Now these are 2 sterile tubes I'm using, so I'm not concerned with that. But you see the problem there. Now, what happens if I ask my lab partner to hold the tube? I'm going to ask the cameraman to extend his hand and we'll see just what might happen. So if the cameraman actually is holding the tube, and I grab the stock and I say, hey, just hold on a second, I'm going to transfer this into your tube. And that cameraman sneezes all of a sudden, achoo, or has tremors, or is just nervous because what I'm doing is I'm transferring live culture, and I miss and I stab his hands. I've just inoculated him instead of the tube. This is a very narrow mouth on this test tube, and there's a good chance you're going to miss it. And the older you get, trust me, the worse your eyesight, the more likely you are to miss. So what does this mean? It means that you and you alone do the transfers. You do the transfers side by side in one hand. You're inoculating loop in the other, and you have full control. The left hand always knows what the right hand is doing and vice versa, so long as those hands are attached to the same body. Okay, so that's the first thing. Now, what we are going to do here first is label. It's always important to have labels. In fact, it's crucial. If you look in your lab manual in session 3, it asks you to take a nutrient auger slant and write on that slant from broth, and then take another one and write from slant, and then write 37 degrees under each of these. I'll explain these labels in a moment, but first I'm going to show you where I want these labels. Don't ever label right under the cap, because if the cap is ever pushed down, it occludes the label. So we want to always label a couple of centimeters down. I usually label along the slant somewhere and along the slant line, and I am just going to write from slant, and I'm going to abbreviate FS. I'm going to write 37 degrees body temperature. On this next slant, I'm going to do the same thing, only I'm going to write FB, which is from broth, 37 degrees. The 37 degrees tells you that that is the temperature we are going to incubate these tubes at. The from broth tells you which stock culture I am going to use to inoculate this sterile tube. The from slant tells me I'm going to use the slant stock culture. I'm going to do this again on these two slants. I'm going to write FS, only this time I'm going to write 25 degrees Celsius, meaning I'm going to incubate this tube at 25 degrees, and I'm going to take the stock culture from my slant stock. So that means over here on this last one, I'm going to write from broth 25 degrees. I'm going to turn the rack around. I'm going to do the exact same thing on these broth tubes. Again, minding that I come down a couple of centimeters below the lowest edge of the cap. One thing I want you to note, don't ever write on the white portion, because ethanol will not remove your label. So we always write on transparent glass when we label tubes. So I'm going to write from slant 37 degrees C, from broth 37 degrees C, from slant 25 degrees Celsius, and from broth 25 degrees Celsius. Okay, so our labels are done. And what I'm going to do, first of all, is I'm going to take the 25 degree tubes and I'm going to move them over here by themselves and put the 37 degree tubes over here, because those are all going in, or those are going at different temperatures. Now, I'm going to take the from broth tubes and put those together, and from slant tubes and put those together. So that means here that I can efficiently inoculate all of those that are coming from the broth stock, and I'm going to do that first. Okay, here I go. Here is my broth culture. Serratia marcescens, I always read the label. I pay really close attention to that label, because when you have multiple stocks of different species in a tub, you have to be careful not to pick up the wrong stock and inoculate the wrong medium with it. Okay, so it says Serratia marcescens. I know what I'm working with. I'm going to flame my loop at the top of that blue cone. You can see it get bright orange. I'm going to, this first time, just flame all the way up the nichrome wire. You can either count to 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You get the idea. To cool the loop off before you place it in the stock. Or in the case of broth stocks, you can simply go in and you can let the liquid cool the loop immediately. I want you to note something. I am not sticking this loop all the way to the bottom of that tube. To do so would contaminate a good stretch of the nichrome wire. Why contaminate all of the nichrome wire if I don't have to? My goal here is simply to bring up a loopful of liquid and transfer it to the tube. Okay? So I'm going to knock that loop off, that loopful off. I'm going to re-sterilize this. I'm going to lay this down for a second, okay? Okay, so this is the broth stock. I'm going to take my first tube, which says from broth 37 degrees. I said that when you work with test tubes, you grab the stock and the new medium in one hand and you grab them by the base at the palm of your hand. You cup them with the palm of your hand. You use the hand that you don't write with to do this. So I'm right-handed. You can see that the caps are loose, which you need. I'm going to take my right hand and I'm going to do this so efficiently that I'm going to grab the furthest cap here between my pinky and my ring finger, and I'm going to grab the second cap between my ring finger and my middle finger, and I'm going to take those off. And then I can easily put them back on using my thumb for guidance, and I can take them off again, put them back on, take them off. If you were here right now, you'd be practicing this. So again, I have total control. I'm holding these upright. I never tilt the tubes or the broth is going to spill. So you always keep test tubes upright, and voila, take the caps off, put them right back on. And that helps me prevent contaminants from the air settling into our tubes. What am I going to do with the loop? I'm going to hold it like a pencil. And so I've got it just like a pencil, but that frees up the space I need to grab the caps. So the first thing I'm going to do, flame the loop, count to 10, or in the case of broth culture, just stick it in the liquid and it'll cool down immediately. I'm going to grab those lids, take them off. I'm going to go into my stock culture, grab a loopful of the broth, bring it over onto the surface of this slant, and I'm going to simply smear it back and forth in a circular motion up and down the surface of the slant, not gouging the slant, just on the surface. The bacteria are going to grow on the surface, remember, just like they did on the plates. When you've done this, you immediately bring the loop over to the flame, sterilizing it. You put the lids back on, and you're done. I'm going to put this loop down for a second, and I'm going to grab a bucket. And this is my subculture. The subcultured tube is going to go in that bucket. This bucket, I'm actually going to label 37 degrees Celsius. So once I have inoculated it and it is no longer sterile, I'm going to put it aside so I know I've inoculated it. What am I going to do next? Well, this next tube says from broth 37 degrees, so I'm going to grab this tube. Now I'm going to be transferring from broth to broth. Okay, let's see how that goes. I flame my loop. I grab the lids. I go into my stock. I don't know if you can hear it singe, but I can. So I know when it's cool. Grab the bacteria, and I move that drop of liquid culture over into the other test tube, and I just kind of tap the sides and let the cells fall off. And on the way up, I tap the sides just to make sure that when I come up, I don't have any broth left. And then I move it into the flame, flame it. Now, I didn't go all the way into the broth on either, so I do not have contamination higher up in the nichrome wire. So I'm going to put the lids back on, and this was 37 degrees, so where does this go? Over here. Now, I just flamed this, so it is cooling off. So I'm going to go ahead and I'm going to do my other two. I'm going to grab the lids off. This says from broth 25 degrees, so I'm going to grab a loopful and transfer it directly over. Tap it, flame it, put my lids back on. Now, this one said 25 degrees. So I need a separate tub for that. I'm going to move this slant stock out and put it in here. And what I will do is I will label this tub 25 degrees C. That's going to stay at room temperature. So we've got 25 and 30, 37. Okay, I have one more tube that says FB, from broth. So here, again, I'm going to flame my loop and grab the lids. I grab the lids off of these two. I go into my broth culture. Here it's Singe, it cools down. I grab a loopful. And now, what do I do on the slant? On the slant, I simply spread that liquid across the surface. And now I've inoculated it. I flame my loop, put my lids back on. Now, this goes in the 25-degree cup. And I am now done with the Serratia marcescens broth culture. I'm going to put it out of the way, and I'm going to grab our other stock culture. This one is on a slant. When you grab bacteria from a slant, the loop doesn't immediately cool down like it does in the broth. So you either need to count to 10 seconds, or you need to do a little trick I'm going to show you. Actually, I'll show you without doing that transfer. If you take a hot loop into a slant culture, you can push it down between the medium and the glass, down into that sterile medium, and then it cools it down, and then you can grab the bacteria off the surface. Now, when students grab from auger slants, our stocks, they like to just grab a ton, and they keep scraping and scraping and scraping, and sometimes they even gouge the surface of, and they push the loop down into the auger. You don't want to do this. There is no need to do anything but just tap the surface of an auger slant to pick up enough cells to transfer to inoculate the sterile media. You don't need to see a big chunk of bacteria like that. Remember, this is microbiology. You can have billions of cells on the end of that loop and not see them. So if you just touch the growth, that lawn of bacterium on a stock culture slant, you can pull it out and you can move it over to the sterile medium. And again, all you do is swirl around up and down. I usually turn the loop over too as well, do both sides. Flame that. Put the lid back on. This says from slant 37 degrees, so we'll put it in that bucket. Now this is me going from a stock slant into a sterile broth. So for this same procedure, my loop is cool. I grab some bacteria off the slant and I inoculate the broth, shake those cells off, tap the excess liquid on the glass side, flame my loop, put my lids back on, and what do I have? 37 degrees right there. My last two subcultures. It says from slant to FS, so I know I'm taking it from the right stock. This one's going to be incubated at 25 degrees. I don't need to reflame my loop because I just did, and it's nice and cool. I tap, grab some bacteria, transfer it into the broth tube. I'm going to put my caps back on and flame. It's okay to put the caps on before you flame. I usually flame before I put the caps back on. Okay, put that in, and our final culture transfer is again from slant to a slant and incubating at 25 degrees. I'm just going to show you my little trick again. I'm going to flame this loop, get it red hot, take off the caps, and I'm going to push it down. I don't know if you heard that sizzle. Sounds like steak on a grill. And I'm going to tap there, and then I'm going to move it over to this tube. And there we go. Flame it. Put it down. Did you guys notice what I did? When I transferred that, I missed the tube. I was showing you and I had it bent like this so I couldn't see the mouth of the tube. So what I did is I inoculated my finger. I felt it touch my finger. So let's use this as a learning example. So what did I say you do? I spilled. What did I spill? I spilled Serratia marcescens. On what? On my finger. Did it get anywhere else? No. So it's localized on that finger. What should I do? First of all, I'm going to put this slant in the 25-degree tub. I've got Serratia marcescens on my finger. And so I need to immediately clean it up. But who, if this happens to you, who tells you how to clean it up? I do. So I'm going to tell you, okay, what you need to do is you need to go over and you need to wash your hands, okay? So I'm going to use a little soap first, and I'm going to wash that off. Now, that's not a typical hand wash, is it? If you know you put billions of cells of something on your hand, you don't want to just use the mild hand soap. What should we use? Do you recall that we have hand sanitizer for special circumstances such as spills? So at this point, I would tell you, okay, now use hand sanitizer and let that air dry. Okay? So I did this on purpose so you would see a mistake. This is what I want you to do when you have any accident at all. Don't be an ostrich with your head in the sand. Don't try and cover it up in some surreptitious, clandestine thing. You have to yell, "Marla, I spilled," and I will ask you what you spilled and we'll take care of it. And it won't be a big deal. Okay? All right. So now we've got our cultures transferred. I'm going to turn off our gas because when you don't need the gas, it needs to be off. Once I've done that, I can remove my goggles. We have to put our cultures away. And we have two buckets. One that says 25 degrees. So I'm going to leave that out on the lab bench and we will check on that in session 4. And I'm going to put the 37-degree cultures back in the incubator. Let's meet back at the incubator and see where I place these. The screen that it is indeed 37 degrees. I'm going to place our bucket in the middle of the shelf. We close the door rapidly and we're done. So 25 degrees on the lab bench, 37 degrees in the incubator. We are good to go. Okay, now let's meet at the goggle cabinet. Okay, you haven't seen this side of the classroom before, but in our lab in the front left corner, we have an electric goggle cabinet, and it actually sterilizes our goggles between use. So whenever you remove goggles from this cabinet, they are sterile. They may be dirty, and you may need to use lens paper to wipe them clean, but they are definitely sterile. So when we put the goggles back, we put them back the way we found them. And you'll notice that all the goggles are sitting nicely upside down on their lenses in this controlled cabinet. And we want to make sure that is always the case. Then when everybody's got their goggles in there, we can close the cabinet. We turn the dial to 15 minutes, and ultraviolet light disinfects those goggles over the next 15 minutes. Okay? So you can be assured when you get goggles out in this class, they are already sterile. Okay. Let's meet back at my lab bench and finish the cleanup procedure. Always at the end of every lab, you are left with stock cultures. Stock cultures must never be left out on the lab bench. You never walk out of the room with live bacteria on your bench. So what we need to do first is you need to take paper towel, a little squirt of ethanol, and always wipe the labels off the stocks. So we wipe the labels off the stalks, and then follow me over to the edge of the room. We keep the stalks in a rack or a tub when we carry them. Do not carry the stalks in your hand. So we are going to walk them over to the side of the room. And here we have our microbiology waste disposal area. There is an area for glass slides. There is an area for broth tubes only. This middle area is for our Petri dishes, which are plastics. They still need to be autoclaved and then thrown away. And then we have a rack for agar tubes only. Everything is labeled. It's very clear. Today, we have a nutrient agar slant stock of Serratia marcescens. We have removed the label. It is time to place it in this agar tubes only slot. Over here, we have an area for broth tubes. Here's our Serratia marcescens stock culture. Where the label has been removed, we place it in the broth tubes. Throughout the semester, I will bring you over to this area again and again. Disposing properly of microbiological waste is paramount. One thing that I want you to know is we never, ever, ever put anything down the drain in this class. No live cultures ever go in the drain. Even if it's just, even if it looks like clear broth, because you've inoculated it and you inoculated it wrong, it's not sterile anymore. You inoculated it. So if it's a mistake inoculation, it still has to come over here. All right, let's meet back at the bench and summarize today. So it's time to wrap up. Let's just review very quickly what we've learned today. We went through a number of lab safety procedures. They included always remain seated when you are inoculating or working with live cultures. When you're using an open flame, you must have goggles on, even over your glasses. You must have your hair pulled back. When you're working with inoculating loops or needles, never hand them directly to anybody else in the class. Always make sure that you put them down on the bench and let that person pick them up. Remember, you do not pick up test tubes by the caps, not ever. When you are doing transfers, you hold both the stock and the new medium tube in your hand to do the transfer. You never let anyone else help you with the transfer. It's all you and no one else. You dispose of stock cultures by wiping off their labels and placing them in either the broth or the agar tube disposal area in the classroom. Remember, always keep your hands away from men, mouth, eyes, and nose. We worked with Serratia marcescens today. Serratia marcescens is on the screen behind me, spelled out for you, S-E-R-R-A-T-I-A M-A-R-C-E-S-C-E-N-S. I expect you to be able to say Serratia marcescens. The T sounds like a sh, Serratia marcescens. And know how to write that, be able to spell it correctly. If you are handwriting it in cursive, that's fine. If you are going to print it, it must be underlined, and make sure those underlines are separate, not connected. And the genus is capitalized, the species is completely lowercase. We will check the effect of temperature on colony morphology and phenotype when we return and see if it makes any difference at all for this organism. I think that about wraps it up. Don't forget, aseptic transfer means flaming your loop before and after you complete the transfer. I'll see you next time.